हाँ जी, अस्सलाम वालेकुम everybody. चलिए जी, बाकी भी आ रहे हैं. Let's just wait for. Sorry, I got 2 minutes late. Okay. Right. चलिए जी, अस्सलाम वालेकुम once again for those of you who have joined. All right, चलिए जी, अस्सलाम वालेकुम finally once again. I hope all of you are well and enjoying the lovely weather in Pakistan. लिहाज हमें आपका नहीं पता, लेकिन और में finally it's cold weather here, alhamdulillah. चलिए जी, so we have planned the new topic today, but I really want to go through the questions, especially this one. This question that I said for homework. So let's just quickly for the first 5 minutes, let's go through this, inshallah, और फिर हम अपने आज new topic की तरफ आते हैं, which is the enzymes. चलिए जी, Bismillahirrahmanirrahim. Let's begin. So, do you mind if I ask you, did you do this homework? मैंने पता नहीं आपको एक PDF भेजा भी था कि नहीं भेजा था. चले बहरहाल. So let's see what this question is. It says starch is a carbohydrate stored inside plant cells. Okay. Explain why starch is more suitable storage substance than glucose. Well, for this, we need to know some details about starch and some details about glucose. So let's quickly brainstorm and see if we know some things about starch. So starch we know is a large, complex, and it's insoluble. Okay. And glucose happens to be small, okay, and not complex. It's a very simple molecule, okay, and it is also soluble. So now let's look at the question once again. Well, can I explain why starch is more suitable storage substance? Okay. So if we need to store the carbohydrate, why should it be in the form of starch and not in the form of glucose? Well, the answer is very simple. If that's a cell, okay, and that's the nucleus, if there is glucose inside and not starch, what will happen is that because of the fact that glucose is soluble in water, होगा क्या कि this if glucose enters the cell, the water potential will actually decrease. Of course, as compared to the outside. Oops, I'm sorry. As compared to the outside. And if there is higher water potential on the outside and there's lower water potential on the inside, guess what will happen? The water will keep on entering the cell and eventually the cell will burst. I mean, that destroys all of the idea of storage. Cell ही नहीं रहा तो फिर store क्या करना है? So the reason why glucose is converted into starch is because if you have a cell which has starch stored in it, since it is insoluble, so that does not change the water potential. So therefore, no water is actually going to enter. So it stays as it is. And then when it is required, of course, then it is converted. Okay. So if you look at the marking scheme, there you go. It's a 3-mark question. It says point number 1, it is insoluble, of course, referring to starch, and it's a large molecule. Molecule ko unhone underline kiya hua. Did I mention the word molecule? Of course, I was drawing in terms of a diagram, but of course, you need to mention that it is a molecule. Yeah, complex hai. Yeah, I did mention the word complex. It remains inside the cell, cannot cross the membrane, does not change the water potential. That's the main point, bachcho. Does not change the water potential and therefore prevents either gain or entry of water, of course, by the process of osmosis. Okay, I hope everybody understands this. And by the way, this is an often asked question, so you should know this, how to answer this. Let me also read what the examiner has to say about this. Very few candidates linked storage molecules to the concept of osmosis. Many candidates wrote about starch or glucose traveling in the blood and significant number incorrectly describe glucose as a large molecule, as a larger molecule than starch. Halaki starch is bigger than glucose. There were many references to starch and glycogen digestion, which was not relevant to the question. चले जी. Now, the next part that I basically want to come, असल में this question, by the way, बच्चों, is from ATP. The only reason I brought this question up was because I wanted to do this part. Okay. उसने आपसे, he has basically asked you in the question that you have to um, draw. Yes. Now you have to draw the cell. पीछे ऊपर उसने लिखा हुआ, you have to, yes, उसने लिखा हुआ है. A1 देखें, in a space provided on figure 1.3, so this grid is figure 1.3. उसने आपको कहा हुआ है कि draw and label a cell as it would appear after immersion in water for 30 minutes. Okay, तो हमें पता है, वो कह रहा है this nucleus should not be shown. Nucleus उसने show नहीं किया हुआ. अब इस सेल को हमने वाटर में डाला है, in pure water. Now if we have entered this cell in pure water, what this means is that the water potential inside the cell is lower, okay, and the water potential, of course, outside, that's in the water, is higher. So clearly there is a water potential gradient being established. It's higher on the outside, it's lower on the inside. So guess what will happen? What should happen? The water should enter the cell and the cell should become what is known as turgid. Okay. Now the question is, how do you think we are going to draw? Did all of you draw this cell? Yes or no?
Yes.
Okay, चलें. जिसने बनाया है, quickly if you can share the diagrams, एक snapshot खींचे picture और group में भेजें जरा. मैं तो आपको draw कर ही दूंगा, लेकिन I really want, and for those of you who didn't draw, it's okay, take a minute and draw it right now. I want you to draw this cell, okay? Don't draw the grid. I mean, just draw 8 boxes like this, okay? और उन 8 boxes में आप cell को draw कर लें, ऊपर वाले को, and then redraw the cell. I really want you people to do this. Let's see, and for those of you who've done this already, if you can kindly share the picture, not in a private message, in the group, okay? Come on, class, don't shy away. Quickly, for those of you who are done, Fatma, आपने कर लिया, आप जरा भेजें ना, मैं wait कर रहा हूँ, quickly. Niaz, did you find time to do this?
I forgot. My bad.
Okay,
So what are we doing now?
you can do it right now. I want you to redraw the cell once it is immersed in water, the same cell.
Okay.
Go ahead. Come on. Take a minute, everybody. इसको draw करें जरा. Please सब draw करें. Trust me, if you do this question correct, then I'm sure you understand this topic really well. और आप हैरान हो जाएंगे. This question is actually from 2002. It's really a long ago. आप में से पता नहीं अक्सर लोग तो पैदा भी होंगे. Okay, great. ठीक है, शाबाश. Good. Good. Very good. Okay. पूरी क्लास
सर
को,
मेरे पास फोन नहीं है अभी, तो मैं व्हाट्सएप
अच्छा
पे
चल,
करता हूँ.
कोई बात नहीं, कोई बात नहीं. It's okay. That's perfectly fine. For those of you who did send, I hope you don't mind if I show this to the class, your pictures. Okay, so we have this lovely picture here. Wow, that's a good picture, mashallah. And then you also have this lovely picture, which is also a tradie picture, वैसे by the way, लेकिन चले कोई बात नहीं. ठीक है, alhamdulillah, I appreciate. Now, असल में देखें, the reason why they gave a grid is, an incorrect way would be to draw this much big cell. देखिए, why is this incorrect? This is incorrect because at the end of the day, when the cell becomes turgid, the cell wall cannot increase in size. The cell wall has to stay same. So सिर्फ आप slightly थोड़ा सा उसको यहाँ से बाहर निकाल के दिखाएंगे. That's it. And that's it, like this. Okay. And then, of course, you are going to show maybe the vacuole to be slightly bigger, something like this. And that's it. I mean, आई एम श्योर जो आपने ड्रॉ किया है आपने यही जेन में रखा होगा. सो आई मीन आई वांट टू शो यू व्हाट द एग्जामिनर हैज टू से. बच्चों, दिस इज रियली इंपॉर्टेंट. प्लीज आई वांट यू टू अंडरस्टैंड दिस. It says it was expected that the drawing would exceed the grid lines both vertically and laterally, showing that the cell had increased in size. लेकिन of course अब देखिए मैंने भी उसको line से exceed करवाया है, लेकिन there is a limit to which I have done it. I did it. You should not, I mean, imagine me drawing this much big cell. Of course, that's never going to be the case, okay? So it does swell. When something swells, it does increase in size, but of course, we don't have to go beyond limits. ठीक है जी. चलो जी, बाकी क्वेश्चन इंशाल्लाह वो इतना कोई इम्पोर्टेंट नहीं था. ETP में जो हम आएंगे हम देख लेंगे, लेकिन दिस इज बेसिकली व्हाट आई वांटेड टू reiterate the point. अल्हम्दुलिल्लाह. ठीक है, चले जी, अल्हम्दुलिल्लाह. So that finishes our second topic, which is diffusion and osmosis. And today we are all set to begin with our lovely new topic. Again, a very small topic that I am hoping, inshallah, that I will be able to manage it in today's time. So let's see how things goes. Chanaji, Bismillah. So let's begin with our new topic, that is the enzymes. Again, that's our third topic. ये जो पहले तीन topics हैं बच्चों, all of these topics, they really set precedence for your entire syllabus, because all of these topics, we are going to talk about enzymes, cells, and diffusion osmosis virtually in every topic. So please pay attention. Okay, now to begin with, let me select a good color. Yeah, I think a green color would do. So before I jump on to the basic definitions, I just want to make you guys understand uh, that a typical chemical reaction, I mean, what we call a thing as a chemical reaction would look something like the following in the dynamics. A plus B gives you C and D. And we've discussed this already. कि जो A or B हैं, इनको we can call them as substrates, okay? Or more easily, we can call them as reactants. And then, of course, this thing is the products. And this entire thing is, of course, known as the chemical reaction. Now, there is something that can be added to this chemical reaction that will alter the rate of chemical reaction, usually speeding it up, and that is known as a catalyst. So how do we define a catalyst? Catalyst is a chemical that speeds up. Usually आपके level तक हम speed up ही देखते हैं, लेकिन it can alter basically either speed up or slow down. But at your level, it's okay that if you only write speed up. So it's a chemical that speeds up the chemical reaction without being chemically used up. Without being chemically used up. Now I've used this word chemic without being chemically used up. क्या मतलब? Well, what that means is maybe a catalyst is solid at the beginning of a reaction, and maybe it can become in powdered form at the end. This is a change in the physical form, but this is not a change, and it stays the way it was chemically, but it has changed physically. Okay, so again, the condition is it won't change chemically. Now, when this chemical reaction is happening within a cell, when it is happening within a cell, you know that you don't call this thing as a chemical reaction. You call this thing as, come on, who will tell me? When a chemical reaction is happening inside the cell, we call that chemical reaction as a metabolic reaction. Okay. And the catalyst that we use in the metabolic reaction will not be called as a catalyst, लेकिन उसको हम क्या कहेंगे? उसको हम कहेंगे biological catalyst. Since it's happening within the cell, no? Bio means living. Since it's happening within the living cell, we say that it's a biological catalyst. And instead of writing such a big name, we call biological catalysts as enzymes. So if I were to now define enzymes, they are biological catalysts that speed up the chemical reactions without themselves being used up, themselves changing chemically. Okay, so that's a classical way of defining enzyme. Asal mein to you only need to know the definition of catalyst and enzyme, but again, I thought it was appropriate that we set all of this context. Okay, without themselves being chemically used up during the reaction. Okay, now there are some characteristics that we are going to talk about the enzymes. So let me make a web spider diagram if you like. So let's talk about these characteristics. Okay, so the first thing, Bacho, that we need to know about enzymes are that they are made by cells. Yani ki the cells manufacture enzymes, okay? And of course, their chemical nature is protein. Chemical nature is protein. There are proteins in nature. Okay.
Sir,
Yes, bache.
sir, is तरह enzymes tissue है?
No. They're made by cells. Enzymes to bache dekho chemical hai na. It's just a chemical. It's not a. You don't call enzyme as living thing. It's not a tissue. It's just an. It's just a chemical. It's not a living thing. It's made by cells. ठीक है, जिस तरह सेल और चीजें बनाता है और chemicals बनाता है, इसी तरह it is making this chemical which is aiding in different chemical reactions. So it's not a cell, neither is it a tissue, neither is it an organ. It's just a chemical made by cells. Okay. Acha ji. So their chemical nature is protein. They're proteins in nature. All right, and then they have a three-dimensional structure. 3D structure. इसका एक टेक्निकल नेम है, लेकिन वो आप ए लेवल्स में इंशाल्लाह डिटेल में देखेंगे. You don't need to go in that details. And of course, as I said, they remain chemically unchanged at the end of the reaction. Remain chemically unchanged. Unchanged. At the end of reaction. Okay, now a very important point, and that is they have active sites. Now, what exactly is an active site? Let me draw an enzyme and show it to you what an active. So, for example, if this is an enzyme. Oops, I'm sorry, my bad. Okay, मैंने E ही draw कर दिया, but this is an enzyme, and the portion that I am highlighting with yellow color, all of this is basically the active site. Okay, this is known as the active site. ठीक है, अच्छा, अब एक्टिव साइट के कुछ फीचर्स हैं बच्चों जो कि हमें पता होने चाहिए. Active site basically is a cleft or a depression or a groove, if you like, any word that you can use that signifies this meaning. It's a cleft or a depression on the structure of enzyme. ठीक है, और the substrate fits into it. Yeah, every substrate can fit into it. Well, no. That brings us to this point, the next point, which is they are very specific in nature. Okay. What do I mean when I say that they're specific in nature? What that means is they have the shape of the active site is specific. Each enzyme has a unique specific shape, unique slash specific of the active site. So for example, if I were to draw another enzyme, maybe it would look like the following. Pac-Man, the typical Pac-Man. So, I mean, you can clearly see that the shape of this active site, again, that's also an active site. You can up clearly declare that the shape of this active site is very different from the shape of the active site that I've drawn. So it's different from above. Okay. All right. So when I say that it is specific in nature, bachcho, what I mean is the enzyme. So for example, enzyme responsible for digesting protein, for example, for digesting proteins is not going to digest fats. To digest fats. Okay. So यहाँ पे मैं आपको examples भी दे देता हूँ enzymes की. I think it's a good point that I should mention this. You know, बल्कि wait. I think let me add this point here that the names of enzymes end with ASE. For example, amylase, lipase, etc. Okay. Coming back to the sixth point, which was about the specificity, जो fat digesting enzyme है, its name is lipase, and of course it's of a different shape. जो protein digesting enzyme है, and what's the name for protein digesting enzyme? Come on, tell me quickly.
Protease.
Protease, शाबाश. Protease. और जो carbohydrate digesting enzyme है, digesting enzyme, that is known as amylase. चलो इसको भी मैं कैपिटल में लिखता हूँ, बाकियों को कैपिटल में लिखा है amylase. ठीक है, इसके बारे में इंशाल्लाह we will talk about in detail, but as for now I think this is sufficient. Okay, so again these are the specificity in nature. Since this was an important point, तो इसके बारे में मैंने आपको detail बताई है. Same goes for active site. All right, and then point number 8, they're used in small amounts. Used in small amounts. By the way, ये जो features हैं ये एक साथ कभी भी वो आपको question में नहीं पूछेगा. You just need to understand and know all of them. Okay, never you will find a question in the past papers which will say describe or outline or list the characteristics of enzymes. So that's never going to be the case. Okay, and then you have point number 9. Their activity is maximum at optimum pH and optimum temperature. यानी कि एक टेम्परेचर होता है जिसपे उसकी एक्टिविटी बेस्ट होती है, मैक्सिमम होती है. That temperature is known as the optimum temperature, and there is a pH value at which the enzyme is working at its best, and that's known as the optimum pH. ठीक है जी. So these are the characteristics of enzymes that we need to know. Okay, inshallah. And then now the next topic that we are going to talk about is the lock and the key hypothesis itself. क्या टाइम हुआ है जी? 8:27. चलें अपनी हम ट्रेडिशनल 1 minute ब्रेक अभी ले लेते हैं. It's 8:27. I will exactly resume at 8:29. So I'm ending the meeting. Take a 1 minute break. Go get yourself a glass of water. Get up, take a stretch, even if you don't feel like doing it. Stop being lazy. I know it's मौसम lazy होने वाला है, except for Saudi, of course. Inshallah, I will resume in a minute.